Welcome, everyone. Today, we are exploring the fascinating and clinically vital world of microbial pigments. When we grow bacteria and fungi in the laboratory, many of these microorganisms produce striking, distinct pigments. It is tempting to view these colors as mere biological oddities, but in medicine, they serve as nature's warning labels or identification badges. Much like a brightly colored poisonous frog warns predators in the wild, these vivid colors are immediate signals that help us identify dangerous pathogens. To understand how they behave, we categorize microbial pigments into two groups. First, we have water-soluble pigments, which dissolve in liquids and spread throughout their environment. Think of dropping food coloring into a glass of water. These pigments can stain growth media, change the color of infected tissues, and even color a patient's wound dressings. On the other hand, we have water-insoluble pigments, which act like permanent paint. They remain locked within the microorganism cells, giving the growing clumps of microbes, or colonies, their distinctive colors, without bleeding into the surrounding areas. Let's dive deeper into why these colors are so critical to clinical practice. In a clinical setting, microbial pigments are far more than decorative features. They have profound medical significance. Primarily, they serve two crucial purposes. First, they allow for quick identification. Before we can run complex, time-consuming genetic or biochemical tests, a pathogen's distinctive color can provide doctors and laboratory scientists with rapid, preliminary clues about exactly what microorganism they are dealing with. Second, and perhaps most importantly, these pigments are frequently virulence factors. In other words, they are active biological weapons that microorganisms use to survive, invade, and cause disease in the human body. As we will see throughout this presentation, pigments can actively protect a microbe from our immune system, directly damage host tissue, assist the microbe in stealing essential nutrients from the body, and even build a protective shield against antibiotic treatments. Understanding these pigments means understanding how these pathogens attack. Green-blue, Pseudomonas aeruginosa. Our first major pathogen is Pseudomonas aeruginosa, a highly dangerous bacterium commonly contracted in hospitals. It poses an exceptionally high threat to individuals with compromised immune systems, burn victims, and patients suffering from cystic fibrosis. Pseudomonas is famous for producing two powerful pigments that double as lethal weapons. The first is piocyanin, a name derived from the Greek words for blue pus. Piocyanin is a direct weapon. It generates harmful molecules known as reactive oxygen species, or ROS, which directly damage lung tissue. It also paralyzes the lung's natural cleaning system, the mucociliary clearance mechanism, and weakens the host immune cells trying to fight off the infection. The second pigment is pyoverdin, a yellow-green molecule that actually glows under ultraviolet light. Pyoverdin acts as a molecular thief, or siderophore, designed to steal iron from the body's iron-binding proteins. Since bacteria require iron to grow, and the human body actively hides iron from invaders as a defense mechanism, a process called nutritional immunity, pyoverdin gives Pseudomonas a massive survival advantage. Clinically, these pigments provide instant clues. A nurse treating a burn patient might notice the wound dressing has turned a distinct blue-green color, 5:20. Similarly, a cystic fibrosis patient with a chronic infection may cough up green-tinged sputum, 5:20. In both cases, these colors point directly to Pseudomonas aeruginosa, 5:20. Bright red, Serratia marcescens. Next we have Serratia marcescens, which produces a striking bright red pigment called prodigiosin, 5. To remember this connection, you can use the simple mnemonic. Mars is red, and Serratia starts with S, like scarlet, 5:19. However, Serratia has a fascinating biological quirk. It only produces this vibrant red pigment at room temperature, between 25 and 28 degrees Celsius, and completely stops production at body temperature, which is 37 degrees, 5:21. This means that if you grow a patient sample at body temperature, the colonies may look white or pale pink, hiding their identity, 6. If a scientist suspects Serratia, they must grow the culture at room temperature to trigger the production of that red pigment. 6. Serratia marcescens also holds a significant place in medical history. In 1950, the United States Navy secretly released this bacterium over the San Francisco Bay to simulate a biological warfare attack, believing it was entirely harmless. 6. Unfortunately, the experiment was later linked to serious hospital infections, proving the microbe was a pathogen, and the operation remained classified for decades. 6. Today, Serratia is a notorious cause of hospital-acquired infections, particularly in intensive care units among patients on breathing machines or with urinary catheters. 6. Because it is naturally resistant to several common antibiotics, catching it early, such as noticing bright red colonies on a catheter plate grown at room temperature, is vital. 6.20. Golden yellow, Staphylococcus aureus. Let's turn our attention to Staphylococcus aureus, one of the most common and stubborn pathogens we encounter. Its very name, aureus, comes from the Latin word for golden, which refers to its characteristic golden yellow pigment, staphyloxanthin, 7:19. This golden pigment is not just decorative, it is a highly effective physical shield. 7. When our immune system's primary defenders, called neutrophils, arrive to kill the invading bacteria, they attempt to spray them with lethal, bleach-like chemicals and hydrogen peroxide. 7. Staphyloxanthin acts as a powerful antioxidant, neutralizing these toxic chemicals on contact and allowing the bacteria to survive and replicate right inside the very immune cells sent to destroy them. 7.18.21. To prove how critical this pigment is, scientists engineered mutant strains of S. aureus by Dunn aureus that lacked the gene to make staphyloxanthin, resulting in white colonies. 7.21. In animal studies, these white mutants were easily cleared by the immune system and were far less dangerous, confirming that this golden color is a key virulence factor. 7.21. Clinically, it's important to remember that this golden color develops fully after 24 to 48 hours of growth, so young colonies may look pale yellow. 7. Crucially, even MRSA, the dangerous multi-drug-resistant strain, still produces this golden yellow shield. 7. Blue-violet, Chromobacterium violaceum. Now we look at Chromobacterium violaceum, an organism that produces a rare and beautiful blue-violet pigment called violacin 8. This bacterium lives in the warm soil and water of tropical regions across Southeast Asia, Central America, South America, and parts of Africa. 8. Interestingly, scientists synthesize violacin from the amino acid tryptophan, and it has become a major subject of pharmaceutical research because it exhibits antibacterial, antifungal, and even potential anti-cancer properties. 8. However, in clinical medicine, Chromobacterium violaceum is a highly dangerous pathogen. 8. Most infections occur when contaminated tropical water or soil enters an open wound. 8. While infections in healthy individuals typically remain localized, causing minor skin bumps or swollen lymph nodes, the bacterium poses a life-threatening danger to patients with a rare genetic immune condition called chronic granulomatous disease, or CGD. 8.21. Patients with CGD have defective immune cells that cannot produce the reactive oxygen species needed to kill certain bacteria. 8.21. Because Chromobacterium violaceum is uniquely equipped to resist the backup killing mechanisms of these compromised cells, it can rapidly spread throughout a CGD patient's body, causing liver abscesses and fatal blood infections. 8.21. A clinical scenario of purple-violet colonies isolated from a wound after swimming in tropical water should immediately raise a red flag for Chromobacterium violaceum. 0. Orange, red, and black bacterial pigments. Our next slide covers 2 distinct and highly specialized pigments, and 1 pitch black. On the left, we have Streptococcus agalactiae, commonly known as Group B Streptococcus, or GBS. GBS is the leading cause of severe, life-threatening bacterial infections like sepsis and meningitis in newborn babies. To identify it quickly, laboratories grow GBS anaerobically on a specialized medium containing serum and starch, called Granada medium, where it produces a striking orange-red pigment called granadine. To remember this color, think of the famous red-orange terracotta buildings of Granada, Spain. Granadine is also a virulence factor. The pigment molecule itself acts as a beta-hemolysin, breaking down red blood cells, damaging cell membranes, and destroying human tissue during infection. Because of this danger, pregnant women are routinely screened so we can catch and manage GBS before birth. On the right, we have Porphyromonas gingivalis, an anaerobic bacterium that causes severe gum disease. This microbe produces a jet-black pigment through a clever destructive mechanism. It breaks down human hemoglobin from red blood cells to extract essential iron, and then clusters the remaining iron-containing porphyrin molecules together to create its black pigment. When culturing P. gingivalis in the lab, scientists must be patient, as this black color can take 5 to 7 days of growth to finally appear on the plate. Slide 8. Fungal pigments, Cryptococcus and Aspergillus. Fungi also utilize pigments to survive and resist treatment, and their primary weapon of choice is melanin, the very same pigment that colors human skin and hair. First, let's look at Cryptococcus neoformans, a dangerous yeast that causes severe brain infections, like meningitis, in immunocompromised individuals, such as patients living with HIV or AIDS. Cryptococcus uses an enzyme called lacase to produce a thick melanin shield. This melanin protects the fungus from immune cell attacks and significantly reduces the effectiveness of our best antifungal medications, such as amphotericin B. Because of this powerful pigment protection, patients with cryptococcal meningitis require prolonged antifungal treatment lasting several months. To identify this yeast quickly, laboratories use a simple, cost-effective test called birdseed agar, which contains caffeic acid. Cryptococcus neoformans converts this acid into melanin, turning the colonies brown-black within 3 to 5 days, while other yeasts remain white or cream-colored. This is an invaluable diagnostic tool in resource-limited settings. Next, we have Aspergillus fumigatus, a common environmental mold that causes serious lung infections in patients with weakened immune systems or chronic lung diseases. Its airborne spores, or conidia, are coated in a melanin-like pigment that gives them a characteristic blue-gray color. This coating acts as an armor. It prevents immune complement proteins from attaching to the spore, neutralizes the acidic environment inside immune cells that would normally digest them, and shields the spore from ultraviolet light damage in the environment. In animal studies, white mutant strains that lacked this pigment were far less dangerous, demonstrating once again that pigment is a key driver of virulence. Fungal pigments. Tallaromyces marnifi. Our final fungal pathogen is Tallaromyces marnifi, formerly known as Penicillium marnifi. This organism is of immense clinical significance as an AIDS-defining infection in Southeast Asia and southern China, meaning its presence indicates severe, advanced damage to a patient's immune system. Tallaromyces marnifi is a classic example of thermal dimorphism, meaning it has two forms depending on the temperature. When grown at room temperature, which is 25 degrees Celsius, it grows as a mold and produces a striking, bright red pigment. This is a water-soluble, diffusible pigment that bleeds out into the surrounding agar, staining the entire culture plate red. However, when the fungus enters the human body at 37 degrees Celsius, it changes its entire structure, transforming into a yeast that multiplies inside our immune cells. This combination of temperature-induced shapeshifting and the highly visible red pigment at room temperature makes identifying this dangerous pathogen relatively straightforward for laboratory technicians, allowing for faster diagnosis in critical patients. The Runyon classification. We conclude our pathogen tour with Mycobacteria, a specialized group of bacteria that includes the causative agent of tuberculosis. When dealing with non-tuberculous mycobacteria, we classify them using a system called the Runyon classification, which is based in large part on how and when they produce pigments. A simple memory trick to master this system is photo, scoto, non, fast. Let's break down these four groups. Group 1 is the photochromogens. These organisms only produce a yellow pigment when they are exposed to light. If grown in the dark, they remain completely colorless, but turn yellow once exposed to light. A key example is Mycobacterium kansasii, which causes a lung disease very similar to tuberculosis. Group 2 is the scotochromogens. The prefix scoto comes from the Greek word for darkness, indicating that these organisms produce their yellow-orange pigment in both light and complete darkness. An example is Mycobacterium scrofalaceum, which causes swollen neck lymph nodes in young children. Group 3 is the non-chromogens, meaning they produce no pigment under any conditions. Important examples in this group include the Mycobacterium avium complex and Mycobacterium tuberculosis itself. Finally, Group 4 is the rapid growers. Unlike the slow-growing mycobacteria that can take weeks, these organisms grow very quickly, forming colonies in less than 7 days, regardless of whether they produce pigment. An example is Mycobacterium fortuitum, which typically causes skin and soft tissue infections. Summary and key takeaways. To wrap up our discussion, let's review the core take-home messages regarding microbial pigments. First, remember that microbial pigments are not just pretty colors or metabolic waste. They are active biological weapons that help pathogens survive, steal nutrients, and cause severe disease. Second, these pigments serve as vital, rapid preliminary diagnostic tools, allowing laboratory technicians and physicians to identify dangerous pathogens days before slow molecular tests are completed. Third, remember the behavior difference. Water-soluble pigments diffuse outward, staining tissues and wound dressings, while water-insoluble pigments stay locked within the colony. Finally, temperature-dependent pigment production, seen in pathogens like Serratia marcescens and Talaromyces marnefi, is critical for accurate identification in the lab. Ultimately, understanding these pigments gives us deeper biological insights into why certain infections are so incredibly difficult to treat and how they behave in our patients. Thank you for your time, and I am happy to open the floor to any questions.